And Dr. Paluzzi, you're not hearing any chimes on your end with any, like, no excess noise or anything?
No.
Okay, perfect. I know Rob should be here shortly, and then he can help introduce you to everyone. Oh, there he is. Perfect.
Hi, Jean-Paul. How's it going?
Hey, Rob. How you doing?
Good. Long time no see or talk, I guess.
Yeah, it has been a while, hasn't it?
Yeah, it has. So, Megan, are we okay? Everything's set up?
Yeah, everything looks good. I think the YouTube link is working.
Okay. Perfect.
And let's see, what time are we at? 11:59. Maybe wait 1 or 2 more minutes and let more people come. I know there's quite a bit more that are expected.
Sounds good.
Rob, I was just saying earlier, I'm going to cut my video just while I present because my kids are home and one's eating lunch, the other one's going to go up. And so just in case somebody walks in, don't pull one of those.
No, it's okay.
And then for the question period, of course, I'll turn it back on. And so,
yeah, of course. No problem.
So you can tell the people that are on campus in the lab versus those who are nice, snuggled at home, comfortable.
Yeah. Well, some of us have the luxury of working from home, but
yeah,
I really feel for the students because they need to be in the lab and gathering data. But as long as they're safe.
That's right.
Yeah.
All right, well, it is 12 now, and I think quite a few people are funneling in. So I'll just continue to watch that and admit people, but to keep things on time, feel free, Rob, to introduce our speaker for this week.
Yeah, of course, I'd love to. Okay, so we're excited to welcome Dr. Jean-Paul Paluzzi, who's an associate professor at York University in the Department of Biology. Now, Jean-Paul completed his bachelor's and PhD at the University of Toronto, Mississauga, in Dr. Ian Orchard's lab. We actually went to grad school together, though Jean-Paul was a couple of years ahead of me. And now at York, his lab is focused on blood-feeding arthropods, including mosquitoes and ticks, and aims to unravel the complex neuroendocrine regulatory mechanisms that promote various biological processes such as feeding and development. His research group employs an integrative approach that combines a number of cellular, molecular, and physiological methods. And we're excited that he's offered his time today to speak to us today about his lab's recent work. So with that, I'll turn it over to Jean-Paul, and he'll take it from here.
Great. Thanks, Rob.
Yep.
So if everybody doesn't mind, just to keep on time, if you have questions, please save them till the end. I just want to make sure that I do finish in time because I tend to go into a lot of explaining with answers, etc. And just shut off my video now and go ahead with the presentation. So thank you for the invite and for having me. And it's great, although it's obviously nicer to get in contact directly with people and give these seminars and visit and find out what everybody and the institution is like. But understandably, under the conditions, virtual is the best we can do right now. So I'm going to be talking today about 2 of the several stories that we're working on in the lab and discussing their importance in regards to physiological processes in the model organism mosquito we're studying today. Okay, so first let me start off by just kind of giving you a little bit of an introduction. And my lab works on comparative neuroendocrinology. Most of the animals that we work on are insects. But we do also work on ticks, And so we're interested in studying the hormones, their cognate receptors, and not only looking at the structure or the expression of these, but ultimately identifying what is it that these neuroendocrine circuits are actually responsible for mediating, whether it's a behavioral or a physiological process. And so the examples here that I'm showing on this slide are various different types of behaviors or physiological processes that are governed by hormones and their receptors. So on the top left, we have a mosquito excreting as it's blood feeding. On the bottom left, we have a cicada undergoing ecdysis from its old cuticle. On the top right, a metamorphosis of a lepidopteran. And on the bottom right, a blood-feeding hemipteran, a kissing bug as it's known, undergoing cuticle plasticization to allow for the enormous blood meal that it imbibes with each blood meal. So what I like to do is kind of give an analogy, a simple analogy to help understand when I'm talking about systems, and in particular systems involving a distinct hormone ligand and its receptor. Each hormone can be represented by a key, a receptor target as the lockbox, and the eventual processes and behavioral mechanisms that we aim to elucidate. In this case, might be the progression through the life cycle of the animal. Another key, different lockbox, unique and specific for their coupling, mediating other types of behaviors, in this case, mating between conspecifics. Yet another now involved in satiety or in blood feeding or feeding in general. And finally, another set of hormone and receptor pairs that might be involved in some form of homeostasis, whether that's energy or hydrominor balance in the animal. And so the model that I'm going to be focusing on today is the mosquito Aedes aegypti. It's an important model to study, but it's not an ideal model. And what I mean by that, of course, it's not the trackable genetic model like is Drosophila melanogaster. But certainly with recent technologies that have come out, it's getting there. So it's becoming an easier model to utilize, but still nowhere near, of course, things like mice and Drosophila or C. elegans. Why is it important to study this animal? Well, it's a vector of several diseases that ultimately, or pathogens that ultimately cause disease in humans. It, together with other anthropophilic mosquito species, are vectors leading to about 300 million infections globally. And while malaria has been taken care of quite substantially over the last several years, there's still close to about 1 million deaths annually. So in a year where we don't have this bloody pandemic, they are probably the deadliest animals on Earth. But of course, this year there's an exception because we do have this global COVID pandemic. So the graph on the bottom is just showing you the distribution of Aedes aegypti, and over half of the countries on Earth are inhabitable by this species. And in every one of those areas where it is present, it can carry at least one pathogen leading to disease. But what this model is actually showing us is that, while the blue is in cases where there's only a single pathogen carried by the animal, most cases are other colors. So you see green, 2, 3, and 4 up until the red, which is 5 different pathogens that can be vectored by that one species in a particular geographic area. So there's enormous potential of serving for a key vector of disease and obviously having an impact on human populations. So today I'm going to be talking about 2 main stories as they relate to the neuroendocrine control of physiological processes in this animal. The first is the functional roles and characterization of the ancient heterodimeric glycoprotein hormone signaling system. And this is work that was carried out by a former PhD student, David Ruckel. And the second story I'll be talking about is the hormonal control, and specifically the antidiuretic control of the renal tubules. And that's being work that's been carried out enormously by a current PhD student in the lab. So first, the ancient heterodimeric glycoprotein story. First, let me begin by just kind of giving you a very brief introduction without giving you too much additional background. But heterodimeric glycoprotein hormones, as they're well known, involve hormones that we probably all have come across and studied, whether directly or in the classroom. There goes the switch again. Here we go. So of course, the gonadotropins such as FSH, LH, and chorionic gonadotropin are important mediators in regulating reproduction and reproductive related processes. Thyroid stimulating hormone, of course, being an important hormone in governing energy balance, as it stimulates the thyroid, and thyroid hormones then governing metabolic rate in the animal. Of course, these are just some of the main functions, but there are other functions that are known to be associated with these hormones. But one of the important features to denote and characterize and bridges all of these hormones is that firstly, they're glycosylated subunits, and they're two distinct subunits. So the alpha subunit is common within each of these different heterodimeric glycoproteins that you see on the screen here. But the beta subunit is unique to each of these different heterodimeric glycoproteins. And the beta subunit has actually been shown to be very important in conferring receptor specificity. And so that's just shown here, common alpha subunit and multiple unique hormone-specific beta subunits. So think back about 20 years ago, the first iteration, I think it was January, February of 2000, when the first iteration of the human genome came out. I think the final polished version, I mean, there were still other iterations, came out somewhere around 2002, 2003. But when that initial data started to come out, these classic gonadotropins had been studied for decades. Well over half a century ago, we've known about FSH and LH and their role in reproductive, and for TSH, its involvement in metabolic activities. But with the genome availability, it was identified that there's actually yet an additional set of subunits, and this is the alpha 2 subunit and beta 5 subunit. And these were actually shown to produce a novel heterodimeric glycoprotein hormone. So they are associated with themselves, do not associate with any of the other subunits. And what was identified was that this novel subunit containing heterodimeric glycoprotein hormone called thyrostimulant was active on the TSH receptor. So now we have in vertebrates a situation where both TSH and thyrostimulant can act upon the TSH receptor. And actually studies showed that thyrostimulant was even more potent of a ligand on the TSH receptor compared to TSH. All of these hormones are actually able to bind through G protein coupled receptors. I'm sure everybody knows what GPCRs are, so I'm not going to go into any of those specifics on that now. But what I do want to point out is the specific feature of the GPCRs of this particular family. And they are typified by having a very large ectodomain, known as a leucine-rich repeat. And this large ectodomain is actually very important in conferring interaction and binding with their large extracellular ligand, right? So, you think about the combined size of these ligands. Each subunit can range between 12 to 15 kilodaltons for the alpha, and between 15 and 20 kilodaltons for the beta subunit. And so that's a relatively large ligand if you compare it to, say, a small neuropeptide that might be anywhere between 1 and 3 kilodaltons. And so that leucine-rich repeat is really important in providing a binding pocket, and in addition to some of the other extracellular regions, such as the extracellular loops of the GPCR. And one other important feature is that this leucine-rich repeat is actually a very large region of the protein. So on average, GPCRs can range from anywhere between 3 and 500 amino acids or so. Of course, this is just a range. There's exceptions to this. But if we look at LGRs, the leucine-rich containing GPCRs, they tend to be very large, 700 amino acids at least. In the case of the mosquito, about 950 amino acids, and over 60% of the protein is actually present in the ectodomain. And so 600 amino acids plus is in that large ectodomain, with the remaining portion forming the normal regions, the normal characteristics of the GPCR. One last thing to point out here before going to the next slide is that while the gonadotropins and TSH tend to be associated mainly with vertebrates, thyrostimulant is ancient. It's found in all bilaterians, so all organisms, all metazoans having bilateral symmetry. And so it's actually a very highly conserved and very ancient hormone system found in many different metazoan organisms having bilateral symmetry. So what's the function of this system in invertebrates? Well, from previous studies, there's been a suggestion of an involvement in development. There's also been a suggestion of involvement in hydromineral balance. And this comes from studies in Drosophila and as well as the mosquito Aedes aegypti. The expression of the receptor transcript. So here looking at the transcript expression by quantitative PCR, and there are two different studies here that I'm highlighting in the fly on the left and the mosquito on the right. You can see that the receptor transcript for thyrostimulant or the glycoprotein hormone is found in to be expressed and enriched in transitional stages of development. So on the bottom left, we see different stages from embryo, larva, pupa, and then finally the adult stage. You see the transitional stages between larva and pupa adult. You see increased expression with pronounced expression in the adult stage. And similarly, on the right, in the mosquito, you can see that there's a significant enrichment in the adult stage. So definitely constitutive expression, but it looks like there's an enrichment during transitional stages and in the adult stage of the animals. In addition to this developmental expression profile, it was also noted that the receptor is differentially expressed in organs that are known to be involved in hydromineral balance, and that meaning iono or osmolar regulation. So in the fly, it was shown that there's significant enrichment in the hindgut, but there's also expression in the Malpighian tubules denoted by MT, the salivary glands as well. And in mosquito, we also have prominent expression in hydromineral balance associated organs. Okay, so again, here the hindgut, the midgut, and the Malpighian tubules. And so from this, we wanted to study and further identify, well, okay, we see expression that's just correlative, but what is it actually doing in these organs? And so we utilized a technique which I won't have time to explain today, but I will provide you with a paper if you're interested to read up or watch the video when it's available to understand the method a little bit further. But ultimately, what we did was monitor the flux, transepithelial flux of ions across different regions of the gut. So the ileum being the anterior portion of the hindgut and the rectum, the posterior region of the hindgut. And what we did was we applied a recombinant glycoprotein hormone onto these regions and then measured sodium flux across the epithelium. Or potassium flux as shown on the bottom. And absorption is a positive, would be on the positive or the top portion of the graphs, and secretion would be on the bottom portion of the graphs. And what you can see in sodium flux is that there's a decrease in the amount of secretion and with an increase in absorption with the recombinant glycoprotein hormone, ultimately meaning that there's an inhibition of natriuresis, because by maintaining more sodium in the body, you're leading to less removal of sodium by the animal. And then the opposite is true for potassium. So both in the ileum and in the rectum, there's a decrease in the amount of absorption across those gut regions, indicating that the hormone actually is promoting what's called caluresis, or the removal of excess potassium. So as I promised, if you're interested, we've got a paper just out in press now. I think it should be available if you've got a subscription in Jove, where we outline this and other protocols for studying the effects of neuropeptides and other regulators on the excretory system. So the first part of this talk is going to focus then on the work by former PhD student David Rocco, and ultimately to study the function of this glycoprotein hormone and its receptor in the mosquito. So LGR1 expression, localization, and characterization is going to form the first part of this section, followed by the functional elucidation of the receptor by looking at its expression profile, examining the expression localization of the ligand, the prospective ligand anyway, and then finally confirming that both the ligand and receptor interact, and to deduce their signaling cascade, as it were, in the animal. So a few years ago, starting up the lab, of course, spent a whole bunch of money on getting antibodies. And what we learned early that is that LGR1, as has been found in other related homologs for receptors for gonadotropins and TSH, are very complicated proteins to work with. They aggregate when expressed in heterologous systems. They are very difficult to design good antibodies for. I think in total, we designed 6 different antibodies against this target and could only have comfort and reliable results with one of those. And so here's just showing you this one that did work well. What we did was, with all these antibodies, was heterologously express LGR1 in cell culture. We tagged it with GFP so we could monitor expression in the cells. And then utilizing this antibody, look to examine for colocalization in cells expressing GFP. And so the figures on the left show GFP in green and the immunoreactive staining for LGR1 in red, and the bottom panel in C showing the colocalization in yellow with GFP and LGR1 immunoactivity. And the panels on the right are just the controls. So pre-absorption with the antigen and untransfected controls, where we see GFP in the pre-absorbed control, and of course, no expression at all in the untransfected controls. So we then looked at the expression profile using both qPCR and this validated antibody. And because of the time constraints here, I'm only going to be focusing on an area a little bit novel to the field, and that is in the reproductive organs. David was particularly interested in this reproductive function because it hadn't been studied before. We, of course, knew potential role of hydromineral balance. But he saw that there was enrichment in female reproductive tissues, both in primary and secondary organs, as well as in the male reproductive organs. But what was particularly interesting, and if you look over to the right, I don't have a laser pointer, of course, to show you, but I should be able to do this with the cursor here. Notice this discrete immunological staining in the testes, this red area here. Okay, so the sodium potassium ATPase staining was used as a marker just to denote the epithelia in the gonads of the male, but see this regionalized specific expression that is red in the testes. And this was really interesting to David. It was really interesting to me. And so this was an area that we decided to pursue further and understand what it actually meant and what it meant for in terms of the function of LGR1. So based on these studies, it suggested then that there was an involvement of this system at the very least for LGR1 in reproduction. And so we sought to study this in further detail. And so on the right, on your left is the SEM images just showing you the structure of the testes, and the testes are ensheathed by fat body. And then we have a optical section through the testes just to show you the organization of spermatogenesis in the testes of male Aedes aegypti. And so at the very apex, labeled 1 here, would be the progenitor cells, the spermatogonia, which ultimately are responsible for the production of sperm, producing the sperm in the testes. And then as you go from the second marker down to, in this case shown as 17, each one of these are distinct, unique cysts where all of the cells within that cyst are actually developing in synchrony. So they're all at a specific developmental stage of spermatogenesis. So 1 is the least developed, and as you go down to 17, they're going to be the most developed. Okay, and the most developed here being the spermatozoa, which are then finally passed on into the seminal vesicle where they're stored before then being released into the female during mating. So we examined expression of LGR1 within the testes in more detail using higher magnification, immunohistochemistry. And what we were able to find is that LGR1 was expressed within the plasma membrane, and that as you went from early stages of spermatogenesis to subsequent and later stages of spermatogenesis, the amount of staining in the plasma membrane greatly increased to a point where it then became very centralized or polarized to only one distinct region of the developing sperm. And so it ultimately was polarizing in the spermatid. And then in late spermatid stages, we saw this elongation of the immunoreactivity. And so at the time, we weren't sure what this actually meant. We weren't sure where this was actually colocalizing within the developing spermatid. We could see that it wasn't in the head where the DNA is stored, but it was in a region just behind the developing spermatid. And so we utilized an array of different antibodies that we could to colocalize to distinct regions. And I'm just going to show you the ones, obviously, that we were able to validate and show to determine where LGR1 was found in the developing spermatid. And then importantly, what we found was that in the final spermatozoa, we saw no longer any staining of LGR1. So dynamic staining as spermatogenesis is taking place with the eventual loss of LGR1 immunoactivity in the final mature spermatozoa. So let's look a little bit closer at the spermatid. So in late spermatids, as I said, we saw that elongation and that localized expression just behind the head, the nuclear head of the developing spermatid. And so we utilized a variety of different antibodies, and here showing you gamma tubulin. Gamma tubulin is important and plays a role in nucleating microtubules, which then allows alpha and beta tubulin to polymerize the microtubules and ultimately form the axoneme structure, which is the backbone of the flagellum. And so here's just a diagrammatic figure showing you where gamma tubulin is localized. It's this region just behind the head of the developing sperm and attached to the central, and this is called the central adjunct. And because we saw that there was an overlap of the gamma tubulin with LGR1, we thought that, well, perhaps given this close association, that LGR1 might actually play an important role in flagellar formation, right? Because this was an active region where the flagellum was being formed. And so to test this hypothesis, we set out to knock down the expression of LGR1. And one of the problems we had to overcome was that if we chronically expressed a knockdown, or if we made a transgenic animal, this had the potential of being lethal, as it's been shown in other organisms such as Drosophila. Animals that have high knockdown of this system have been shown to simply not undergo the appropriate metamorphosis, and you don't get any viable adults in the end. And so we had to modify our approach in terms of how we would to knock down our target. But more importantly, we had to also ensure that we were going to get at a time when we would actually have an effect on spermatogenesis. And what I mean by that is, if we tried it in the adults, we would already have passed the time when spermatogenesis is taking place. And so we had to get in later stages of the larval stage, because spermatogenesis is actually happening in later stages of the larval stage. And so what we decided to do was apply double-stranded RNA to knock down LGR1 expression beginning at the second larval stage. And so what we did was used a plasmid that was originally described by Andy Fire et al. In over 2 decades ago now, that allowed us to produce double-stranded RNA in bacteria. We grew these bacteria, we induced expression to synthesize our double-stranded RNA targeting LGR1. And then we fed this bacteria containing double-stranded RNA against LGR1 to the second instar larva and the subsequent stages, third and fourth star. We waited for these animals to pupate and then examined expression of the LGR1 in 1-day-old virgin males. From this, we were able to show that the relative transcript abundance, both in larva and in the adult, was significantly reduced in both cases, as shown here in the black bars. And as well, we looked at the immunoreactive staining for LGR1 in the testes. Here's the control, and here's a knockdown animal. And by quantifying the immunohistochemical staining in these regions, we can see that there's also a significant reduction in the amount of immunoreactive staining, suggesting that not only are we getting transcript level reduction, but also a reduction in the protein level of LGR1. So great, we have knockdown. We have knockdown suggesting both at the transcript and at the protein level, but what's the functional relevance of this? Well, we were interested and looked at the males, and were able to quantify the amount of spermatozoa in males, and found that the total number of spermatozoa in the testes was significantly reduced. The level in the seminal vesicle, I mentioned this earlier, this is the storage organ within the male that will test the spermatozoa before it's delivered to the female during mating, as well as the total number of spermatozoa is significantly reduced. Okay, so there is a total number of reduction in knockdown animals. And interestingly, if we look at the anatomy of these spermatozoa, on the left here is the control condition, where we just fed with an empty vector control, and the knockdown here on the right. You can see that the length of the flagellum is significantly reduced in the knockdown LGR1 knockdown animals. And when we quantified this by measuring a number of different flagella from knockdown animals, we see that there's about a 25 to 30% knockdown in the flagellar length. So great, another feature of this knockdown, both transcript and protein, and now an anatomical feature that seems to be important. but does this actually lead to any functional consequence in the animal? So what we did next was wanted to test for this function. We mated control and knockdown males with normal colony bred females that had been blood fed. And then we measured the number of eggs as well as the hatching rate of those eggs from those matings. So when we quantified the number of eggs, we found that there was no difference in the number of eggs being laid by females. And this was to be expected, of course, because the females were control. We didn't manipulate them in any way. But when we measured the amount of hatching from those eggs that were laid by females, we found that the knockdown, LGR1 knockdown matings actually led to a significant reduction in hatching, suggesting then that obviously the LGR1 not only has important feature in terms of knockdown and in terms of anatomical features of the spermatozoa, but that these actually have a functional consequence to the animal. So there's a reduction in normal production of normal and viable spermatozoa. So we next sought to look at the expression profile and localization of the subunit. And so we utilized qPCR again, and here just showing you different regions of the nervous system. So B is for brain, T is for thoracic ganglia, and AG is for abdominal ganglia, and then RT is for reproductive tissues, gut, and then finally the carcass. So what is just supposed to show here is that both the alpha and beta subunit, regardless of male or female, is significantly enriched in the abdominal ganglia. There's also prominent expression in other regions of the nervous system, but it's not significant compared to the other organs. So the main region that these hormones were deriving from would be the ventral nerve cord and the abdominal ganglia of the animal. We had an available beta 5 or a beta subunit antibody, and so we immunologically localized the subunit within the abdominal ganglia, and we found between 2 and 3 immunoreactive neurosecretory cells within the abdominal ganglia, which had these processes emanating into the lateral nerves, and these lateral nerves then fed into these neurohemal structures, which, to give a simple analogy, would be equivalent to the neurohypophysis or the posterior pituitary, which is the release site for neurohormones in vertebrates. We also localized the transcript using fluorescent in situ hybridization. And so here we have the alpha subunit, the beta subunit, and finally both subunits together. And what we can see here is that, again, 2 and in some cases 3 distinct cells are found in the abdominal ganglia in a region that very closely associated with what we saw for GPV5 immunoreactivity, suggesting that, you know, we did find where specifically these subunits were being expressed, and that they indeed were co-localized within the same cells. Obviously, this would be a fundamental importance because they couldn't heterodimerize if not expressed in the same cells. So here's just an overview, just showing you the expression within the abdominal ganglia. All right, so we then wanted to, having validated where it's expressed, we wanted to establish that the ligand could actually interact with and activate the prospective receptor LGR1. And so we utilized an approach that had been done previously in lamprey and in Amphioxus, where designing a tethered construct, which is ultimately making a fused polypeptide that allows for the guaranteed heterodimerization, so to speak, of the 2 distinct subunits. And so here we have a construct that has the beta subunit, which contains the endogenous or native signal peptide, which is important for shuttling this protein to the secretory pathway. It is then placed upstream of the GPA2 propeptide sequence, but between these 2 coding regions is this linker sequence. And the linker sequence also has a histidine tag, which would allow us to monitor expression within the heterologous system. Oops. So ultimately, this just allowed the heterologous system to produce the heterodymic glycoprotein hormone and allow us to then test that at higher concentrations. So what we did was verify that expression was indeed happening. And so we collected the lysate from the cells. We collected the media that secreted, that would have the secreted fraction from the cells. And we found that in transfected cells with a tethered alpha beta subunit, that indeed there was the expected product size. But what we did find as well was that whether it be because of a non-optimal secretory signal peptide or what, the mammalian system was not ideal for secretion for this particular construct. And so a lot of it was being retained in the cell. And for the purposes and simplicity here, I'm just going to be focusing on that particular function in the next couple of slides. But before I go to that, I do want to point out that we did test and verify that the secreted fraction, was indeed as expected, and that when we treated this with a deglycosylation enzyme, PNGase, it was actually deglycosylated. So it was being processed as would be expected of a glycosylated hormone. All right, so in the next portion here, we utilized a commercially available system involving a recombinant firefly luciferase that has a cyclic AMP binding moiety that allows for the detection of near real-time events in terms of signaling. So in this particular sensor, increases in cyclic AMP would lead to an increase in the luciferase activity and an increase in light detected by the system. And similarly, if we were interested in testing a GI-related pathway, we could stimulate the system with, say, forskolin, which acts on adenylyl cyclase, and then challenge that with potential inhibitors to verify whether a particular system works through a GI-coupled pathway to reduce the levels of cyclic AMP. So again, using that lysate fraction, because that was the fraction where we saw the highest amount of recombinant protein, we tested this in the GS pathway. So cells expressing the receptor were challenged with either a M-cherry control, which is just the cells transfected, but with an M-cherry, which is just serving as a positive control for transfection, but a negative control for the functional assay. And you can see here, forskolin that was applied for the positive control in the functional assay did indeed lead to an increase in cyclic AMP levels, but we saw no increase present either in the absence or in the presence of the receptor when we applied the tethered hormone. So this meant that the GS pathway did not seem to be a functional pathway for the system. We then went on to test the GI pathway, and this wasn't just on a whim. The reason we started with the GS pathway was because almost all of the other related systems, and by that I mean the heterologous G protein related systems involving heterodimeric glycoprotein hormones, they almost all signal through cyclic AMP. And so, it was expected almost that cyclic AMP and the GS pathway would be the system and the signaling cascade utilized by the GPA2 and LGR1 system. But instead, when we did a in silico prediction of coupling, we found that the receptor was very highly predicted to couple through a GI pathway and not a GS coupling pathway. And so repeating the assay, but now optimizing and testing it through a GI pathway, we found that in the presence of the receptor and application of the tethered protein, recombinant protein, we found a significant reduction in the levels of cyclic AMP, validating then that the GI pathway is likely an important pathway for mediating the signaling cascade between this ligand and its receptor in the animal. All right, so the next section of the talk, the next story I'm going to be focusing on is the antidiuretic control of the renal tubules, a mechanism to combat desiccation or a strategy to fine-tune excretor activity. And let me first just kind of start by kind of giving you an outline of the excretory system. The excretory system in insects involves two main segments of the alimentary canal, the Malpighian tubules or renal tubules, and the hindgut, which, as I mentioned early on, and I can mention here again, is normally broken down into two main areas: the ileum, the anterior region of the hindgut, and the rectum, the posterior region of the hindgut. Now, the excretory system is controlled by several different hormones, and these can be neuropeptides, they can be biogenic amines, and we have evidence as well that even the glycoprotein hormone might have an effect on the excretory system. And I presented some of that data earlier on. But I'm listing here some of the more relevant ones for the purposes of this talk, so that just to familiarize yourself with some of the terminology for later. So insects have hormones known as CRF, or corticotropin releasing factor related peptides, calcitonin related peptides, or DH31, kinin related peptides. The abbreviation later is going to be CDP, serotonin and amine, and kappa related peptides, which are insect specific but are distantly related to neurometa nu in invertebrates. So all of these are known to target different regions, some acting on the same cells, some acting on the same tissues, but each having unique functional actions in terms of how they handle ions and water and things across the different excretory organs. So within the Malpighian tubules or the renal tubules, there are, in most insects, two distinct cell types: principal cells that are mainly responsible for handling cations, as well as the stellate cells that are present mainly in the more distal regions of the tubules, and these are largely responsible for handling anions. What's interesting here is that obviously it allows for the fine-tuning of movement of osmolytes and electrolytes, but you would expect too that if both types of ions needed to be moved around and recycled, that you'd have the activation of both cell types, right? So principal cells for cations and stellate cells for anions. And another feature to point out is that kappa, CRF, calcitonin, as well as the kinin act on different cell types. Kinins acting on the stellate cells, and kappa, CRF, calcitonin, and serotonin acting on principal cells. So those serotonin is not shown here. This is just a model borrowed from the fruit fly. And so in the next few slides, what I'm going to be talking about quite a bit, and just to show you what the assay is, this is one of the assays we use quite a lot in the lab, known as the Ramsay assay. What we do is in a dish, it's an in vitro preparation where we remove the organ from the animal, place it into the dish into a saline droplet, and then within that saline droplet, we add a stimulant, a peptide, a neuropeptide, a pharmacological agent, some kind of drug or whatever, to try to impede the function of the stimulant. And then we can measure rates of fluid secretion by the tubules. We can measure the composition of that fluid secreted by the tubules, whether it's just simply the pH or the ionic composition, such as sodium and potassium levels. So in this part, focusing on the work done by a current PhD student for our society and looking at the antidiarrheal control of the Malpighian tubules with a focus on today the kappa system, as that is stuff that she's published already. She's looking at the functional roles of kappa-related peptides, a source of kappa antidiuretic hormones in the animal, the deorphanization of the antidiuretic hormone receptor, and a signaling mechanism of the antidiuretic hormones. So very quickly, we published this a few years ago, but the tubules, as you saw in the last couple of slides, are acted upon by various different hormones. And serotonin, CRF, kinin, CDP here, and DH31 are all capable of stimulating tubules. And then when we apply one of the kappa peptides in the animal, we find that there's selective inhibition on these distinct diuretic hormones. So kappa can inhibit serotonin. It can inhibit the DH31, also known as mosquito natriuretic peptide, but it has no significant inhibition on CRF-related diuretic peptide, and also no effect on the kinin-related peptide. We also tested the prospective second messenger for this antidiuretic hormone, and this wasn't just, again, just out of the blue. There was data collected in other systems and other models that have suggested that cyclic GMP might be a good candidate as a second messenger. And what we could show is that serotonin, along with cyclic GMP, and in DH31, is very similar. It phenocopies the effects we saw with kappa, where cyclic GMP can inhibit both serotonin and DH31. No significant inhibition in CRF-related tubules, but interestingly, also a significant inhibition on kinin-stimulated secretion. So I'm not going to be able to focus and discuss any more about the kinin. It's something that we're working on now, but this is a unique cell type, suggesting then that it's a unique antidiuretic hormone targeting this specific cell, because we know that it's not active by kappa. So here's a model that, together with studies previously and our work, we were able to develop a couple of years ago. Serotonin, DH31, kappa, and CRF all acting on the principal cells. And I won't go into the details of the transporters here, but I'm happy to elaborate later. And kinin targeting the stellate cells to elicit its effects on the stellate cells. So it's believed that kappa inhibits both Th31 and serotonin-stimulated secretion, but of course we weren't sure how this actually was being mediated. We had some evidence that cyclic GMP might be involved, and in other systems it had been shown that nitric oxide, cyclic GMP, and protein kinase G pathway was involved in the signaling cascade. So in terms of the source of these neuropeptides and the eventual hormones that are secreted into circulation, so this is just a map showing you the distribution of the peptides, the immunochemical localization within the abdominal nervous system, the abdominal ganglia. And so here in each of the abdominal ganglia here, this is actually taken out of the animal. This is an in situ prep showing you the immunolocalization within the abdominal ganglia. This is just removing the the dorsal cuticle so that you can see the abdominal ganglia in place within the animal. And then these processes mediating through the medial nerve, and then laterally to innervate these structures known as the periversal organs. Again, the PVOs are neurohemal organs where these neuropeptides can then be released into circulation and enact as neurohormones within the body. So in addition to localizing the protein level by immunohistochemistry, we also localize the transcript, again using fluorescent instituted hybridization shown here in red. And when we co-localize the immunoreactive staining with the transcript staining, you can see that it nicely co-localizes as shown here on the bottom in panel H. So the source of kappa antidiuretic hormones is indeed the nervous system, and it is, as we saw earlier, the abdominal ganglia. But as you can see, they are discrete neurons, neurosecretory cells. Remember earlier, the glycoprotein hormones were found in those lateral neurosecretory cells in the abdominal ganglia, whereas the cells here are more medial and could be considered medial neurosecretory cells in the abdominal ganglia. So again, going to deorphanize the receptor using a slightly different system here, using a bioluminescent protein from the jellyfish, Aporia victoria, and the protein here being aquarin, expressing the receptor in the heterologous system, and then monitoring for an increase in intracellular calcium, and that intracellular calcium leading to bioluminescent signal or mediated by aquarin, and then that luminescence can be measured by the multi-well plate reader in the lab. And so we applied kappa neuropeptides onto this receptor and found that it was potently activated by these ligands with relatively low EC50 in the low nanomolar range. I think the total final published value was about 4 to 5 nanomolar, and similar efficacy between both copies of the neuropeptide. At very high doses, we found that a structurally related neuropeptide, pyrrokinin 1, is also able to activate slightly the receptor, but we believe this is just a supraphysiological dose and probably doesn't have any relevance to the animal. And we also recently published that this neuropeptide actually has a function on discrete receptors and is responsible for distinct physiological functions in the animal, unrelated to the functions of kappa. We tested a library of neuropeptides, and this is just a subset of those that were tested, each of these being unique in their structure, being unique in terms of their own specific receptors that they are known to activate. And we found that at the very high dose that we tested here, only kappa were able to significantly lead to activation of the receptor. In terms of expression of this receptor, we found that expression was highly enriched in the renal tubules or the Malpighian tubules, and this was true in both male and female. We did see some expression in other areas, but it was not significant as it was in the Malpighian tubules. And so to confirm where specifically the receptor was expressed, we again utilized in situ hybridization. And so the diagram here is showing you the signal within principal cells shown in red, and the arrowheads here pointing to those stellate cells that have smaller nuclei. And you can see that there's no staining associated with the stellate cells, only with the principal cells. And here on the right is just a control where we applied the sense kappa probe, which would of course not bind to the mRNA. So confirming then that the receptor is indeed in the principal cells and is highly enriched within the Malpighian tubules within those cells. All right, so we've got some functions that we talked about. We've got receptor validated now within the principal cells, and we have some early data then suggesting that cyclic GMP is a prospective second messenger. Well, what about those other candidates that I talked about earlier in terms of nitric oxide synthase and protein kinase G? We wanted to study these further, and so we used some pharmacological agents that were available and block these independently to try to discern what the signaling cascade might actually look like in the antidiuretic scheme. And so first blocking nitric oxide synthase with L-name, we found that L-name had no effect on DH31, and this would be as expected because, of course, DH31 is signaling through a distinct pathway involving cyclic AMP. When we applied kappa to DH31, of course, as we saw, it's antidiuretic, it's inhibiting secretion, so no difference from the unstimulated secretion. But when we applied the inhibitor, L-name, we saw that we abolished this antidiuretic effect. So suggesting then that nitric oxide synthase was involved in the kappa-mediated antidiuretic signaling. When we did this with cyclic GMP, the proposed second messenger, we found that L-name had no significant effect. And this actually is making sense because cyclic GMP would be downstream of nitric oxide synthase, nitric oxide stimulating nitric oxide, the soluble guanylate cyclase that then leads to the production of cyclic GMP is sensitive to nitric oxide. And so you're bypassing that synthesis by applying cyclic GMP. And so, of course, we see no effect of L-name. What about PKG? So another inhibitor, KT5823, targeting protein kinase G. Again, no effect on DH31, significant abolishment of effect on kappa. So no longer do we see that antidiuretic effect in the tubules when kappa is applied. And of course now, because protein kinase G is downstream of cyclic GMP, even with back applying cyclic GMP, we no longer see that antidiuretic effect because protein kinase G is downstream of cyclic GMP. And of course, by blocking PKG, you get the abolishment of the signaling cascade. So confirming then that cyclic GMP is involved, as is nitric oxide and protein kinase G. So we're now starting to work out in more detail what are the targets of the signaling system. So where specifically is kappa eliciting its effects and what transporters are being affected in the diuretic signaling system. So let me summarize here by just telling you that hopefully I've convinced you to some degree at least that neurohormones and receptors do indeed play key roles in controlling many physiological and behavioral processes in animals. And of course, here are the examples in disease vectors, but certainly similar systems are active in other animals. Neurohormones and the receptors are important in orchestrating behaviors, regulating physiology, and ultimately broadcasting the news. And by that, I mean, they're important in keeping all the tissues and organs within the body synchronized so that, if there's a particular stimulus or a particular challenge to the animal, each of those organs can be synchronized appropriately to meet the demand of the animal. Elucidating hormones might actually be useful for revealing targets for novel therapeutics or for novel control strategies to eradicate pests, whether they be disease-related pests or agricultural pests. And this is really a promising area because in this way, we could target specific GPCRs, ones that are localized and specific for a particular organism, and get around the potential of this broadcast type chemical insecticides that has been used for decades now. And we know, of course, of the drawback of that being that, it's impacting pollinators, it's impacting other organisms in the ecosystem. And so we definitely want to get away from those broad spectrum type insecticides for pest control. So with that, I'd just like to thank, firstly, all the funding that helps keep the doors open and the lights on and the research going, I guess, in the lab. And particularly want to thank the two lead researchers involved in the research that I presented today, David and Farwa, but as well all the students over the years that have contributed either directly or indirectly to this research. I'll end it there with a advert. If you're interested, students, etc., either in a MSc or PhD, here's some information on the deadlines, the start dates, etc. Reach out, all my contact information is there. Thanks a lot.
Awesome. Thank you so much, Dr. Paluzzi. So we will open up for questions. Anyone who has any questions, feel free to speak up and unmute yourself. But if you're not comfortable and want to write in the chat, feel free to do that. We can monitor those as well. So yeah, any questions, feel free.
Megan, maybe I can ask one first.
Sure.
Get the ball rolling.
Yeah.
So great talk, Jean-Paul. Really interesting work. In the first part of your talk, you, I think, showed a co-localization of LGR1 with tubulin, if I'm not mistaken.
Gamma tubulin, yeah.
Yeah. So did you ever look at a potential role in vesicular trafficking, maybe how it might be interacting with motor proteins in any way?
No, but absolutely, that's something to study more. We didn't have the ability at the time, and you know, it's something probably that we'd have to do through collaboration. But part of the problem is the difficulty in getting good antibodies for that. And what I'm going to try to get going now is utilizing methods like CRISPR and making transgenic animals so it's a little bit more trackable in terms of where specifically expression is, when is it turned on, et cetera, and try to dissect that in more detail. Definitely something to look into in the future, but just haven't done it yet.
And if I could follow up with a second question on the peptides, are these synthetic peptides, or are you expressing them and then administering them to the cell?
Yeah, so the small peptides that I talked about in the latter portion of the talk, those are synthesized by CROs, commercial research organizations, for the most part because they're able to make them efficiently, get them to high purity, and to get them made. I mean, I have a couple of different model heterologous systems. We've got some mouse pituitary cell lines that we can use to produce, because they would normally produce neuropeptides. But the efficiency by which we can get them made is far better through a CRO. And so, yeah, they are synthesized, but they match the activity of the endogenous. There's structurally identical and functionally identical.
Yeah, I guess my question would be if they're folding correctly or if they're functioning properly. But if you're seeing responses, then I guess you can infer that they are.
Yeah, absolutely. And because they're so small, their structure is relatively simple. And so we don't face some of the problems that I guess you would expect with more complicated, like definitely the glycoprotein hormone subunits are much larger and very difficult to get made. You know, we've gotten many peptides made anywhere between, you know, 100 and 300 bucks a pop, depending on sales and stuff. We've gotten quotes for the glycoproteins and it's about $25,000 US. And
wow. So
not feasible to get
cost
them
benefit, right?
Yeah,
totally. Okay, awesome. Thanks.
I have a general question too. I know we have one in the chat, but I will ask, and then if anyone else has questions too, we'll ask. We'll address those after. But in your first sets of experiments, so your LGR1 experiments, perhaps I missed this because I take attendance in the beginning, but can you explain, is there any significance with why you're only looking at males and the male reproductive system? Is there anything in females? Yes, can just explain why your focus is on males in that sense?
Absolutely. So it's an awesome question. And it's something that, you know, I had to let David follow his heart and where he wanted to look at. Part of the reason we stuck with males was because female reproductive biology and the hormonal regulation has been so extensively studied in insects. And we know a lot about how female reproductive processes happen in insects, but there's essentially nothing known on male reproductive biology. There's some early data, but it was an opportunity really to develop something novel and lead. And so that's part of the reason why David decided to pursue the reproductive function in males. We also saw that very discrete staining in the male reproductive organs, as opposed to the broad staining that we saw in ovaries. But absolutely, most certainly, it's an area where actually my postdoc right now is looking at that in another model. And we'll be studying that in that other model, but absolutely something to look at in the future for ADs as well.
Okay, thanks. So one of our questions from our students is, do you expect these results to be generalizable to other species in the Aedes genus or other species in the Culex genus?
I mean, I would say likely, but of course, you know, no certainty there. We see similar expression profiles, even, you know, between mosquitoes and Hyraediptera. And so I would expect that similar expression profiles might be observable, as well as similar physiological functions across these animals. But of course, without having that data, I can't say for sure.
Right. Okay, I have another question. So in your antidiuretic hormone experiments with kappa R, it seems like you're monitoring in terms of output increased levels of calcium, which is what, if I'm understanding correctly, give you the luminescence, which you can then observe.
That's right.
Can you explain the significance of calcium ions in that case?
Okay, so absolutely. So the system we utilize is just, it's a heterologous system. So really it's meant to show interaction and activation between a potential ligand and the receptor. Of course, what it doesn't tell us is how or what is the efficacy or binding specificity, the potency of the ligand within the actual cell, within the actual animal, sorry. And the reason for that is, of course, is that, you know, in the heterologous system, we're overexpressing the receptor. And so it's a very artificial system, right? It does certainly allow us to say this receptor is indeed activated by this ligand. It's not activated by all these other tested ligands, but it can't be directly taken as, okay, EC50 in heterologous system is nanomolar. That's going to be the dose in which is relevant for the animal as well. The other, to answer the calcium question, calcium is used because it's the signaling molecule required for a quern luminescence or quern-based luminescence in that particular expression system. But even if a receptor is not normally coupling through calcium, we have some accessory proteins that we can co-express. Essentially, they're called promiscuous G proteins. And they allow for any receptor, so they say, to be shuttled to the calcium pathway.
Okay. Okay, thanks. Is there any other questions for anyone? If not, I have one more. So this is kind of maybe more of a general question, but I'm just interested in for my own research. So is Alname, is that just a known inhibitor of nitric oxide synthase? Or
I didn't
what?
get the first part. Is it a what inhibitor?
Is Alname, that was your inhibitor that you
Yes.
used? Is that
Yes.
just a known inhibitor for nitric oxide synthase?
Yes, it is. Yeah.
Okay.
Yeah.
Okay. All right. Thanks.
Okay.
All right. Well, if we don't have any more questions, thank you very much for spending time and coming to our biology seminar series. We greatly appreciate it.
Great. Thanks so much.
I echo what Megan said, and we can all give you a virtual clap, I guess, for the presentation.
Thanks, guys.
Thanks so much. And
everybody be safe. And thanks
All
for the
right,
opportunity to
well,
talk to
thank
you.
you.
awesome. Take care.
Okay, take care, everyone.
Okay.
Thanks, Rob. See ya.
Yeah, no problem. See ya.
See you in a couple of weeks.
Yeah, totally. Bye.